this is where we left off yesterday, guys. We're talking about the fields of view, okay, for the different lenses on our microscopes. All right. So if you didn't get this diagram down yesterday, make sure that you have it. Here's the explanation of it. All right. When you look through the scanning power lens, that's the one with the least magnification with the red band around it, you will be seeing an area that is 4,000 micrometers across. All right. 4,000 micrometers is 4 millimeters. All right, so just a little bit less than half a centimeter is the area you can see through your scanning power lens. All right, for a microscope, that's a very wide field of view, believe it or not. All right, the reason we want to have that wide field of view, it enables us to find our specimen very easily because we see a large area all at one time. All right, once we find our specimen, where do we want it to go? Right, into the middle on the end of the pointer. Okay, so you move the little stage wheels around. Okay, just had a brain fart there. Okay, the stage wheels around until it's onto the pointer. Remember that when you're looking through a lens system, that's reversed. Okay, so if the stage is moving to the right, things are going to be moving to the left in your field of view. Okay, because everything gets reversed when it gets refracted. Your low power lens, when you look through that, the low power lens field of view is 1,600 micrometers across. All right, so 1.5 millimeters roughly. Okay, very, very small. Things will appear a lot bigger when you're looking through that one. Okay, and then even more so when you get to the high power lens, your field of view is very small, but what you're looking at is greatly enlarged. All right, the field of view on your high power lens is 400 micrometers. All right, so you're looking at an area that is less than half a millimeter. All right, all the way across. All right, so things will appear greatly enlarged on there. Why do you need to know this? You need to know this because of how we estimate the size of an object that we view under the microscope. All right, it's important when we're communicating to other people about the things that we looked at that we communicate how big they are. Now, that's not ever going to be exact. It's always an estimate, but here's how you do it. If I am looking through my high power lens, okay, at an object, and it appears this big, and I know that the distance from here to here is 400 micrometers, how big is that object? Roughly. About 150, 180, something like that, right? It takes up maybe about a third of that distance, all right? And that's how we estimate it. We go, well, about 3 of those would fit across. Okay, so 400 over, okay, divided by 3, whoop, and we would have our approximate size of the object that we're looking at. All right, so anytime you want to estimate the size of an object, okay, and I think actually I have this on the notes. Okay, estimating the size of an object, this is what you do. Okay, you estimate how many of the objects would fit across the field of view and divide the field of view by that number, which is why we give you the fields of view so that you can do that accurately. All right, now, what if I'm looking at this thing, but it's oriented this way? Does my estimate change? No. Okay? The convention is this. We always measure the longest dimension of the object. Okay? So if you have something that's very oblong, like this, or like a paramecium, which is one of the things you're going to look at today, you always measure it on its longest dimension. All right? So, because obviously, if I measure it along here, I would say that that object's maybe only about 50 micrometers, because a lot of them would fit across oriented that way. But we always measure on their longest axis. Not that you could do that on the microscope, but okay, gives you some idea of what to do. Everybody follow there? All right, so that's what we're going to be doing. We're going to be looking at a few things, okay, under the microscope today, and drawing lab diagrams of them, which is what I'm going to go over next, and then we'll have a little lab report on the microscope and things like that. So it's pretty straightforward stuff. You're only going to be looking at 4 things, and they're all easy to find, so we don't need too much time in there. All right. Right, so we've got that down. That's important. Okay, we're going to skip those for now, and I'm going to hand this out to you. First, I'll pause. All right, listen up here, guys. So you've got your diagram there, okay, your example of a proper lab diagram. So a couple of rules on drawing a proper lab drawing. Okay, first off, they're really, really anal about this at university. So you really want to do this right. Okay, take it from a guy who got a 0. Okay, the first time he did one of these, they're really anal about this. Okay, because I'm not a good artist. I can draw lab drawings because you don't have to be a good artist to do that. It doesn't hurt. Okay, but you don't have to be. But if the format is in any way not the way they want it, they just give you a 0. All right, that's the way it works, unfortunately. So first and most important rule of drawing a lab diagram: they're always drawn in pencil. Always. Okay? You never draw a lab drawing in pen. You always draw it in pencil. All right? And they should take up the whole page. It should look like this. Okay? This rectangle that's on the sheet that you have represents a sheet of paper. You don't have to draw a rectangle around yours. Okay? I see that happen all the time. The rectangle represents the borders of the sheet of paper this was drawn on. Okay? So you get an idea of how much of the page the drawing should take up. All right? What do you notice about all the labels? Yeah, well, they all go to the same side. It usually is the right, but it could be the left as long as they all go to the same side. They don't want them going all over the place because then the lab drawing starts to look disorganized. All right, so they all go to the same side, and you'll notice that they're all straight lines going out from the diagram. All right, if need be, if you have two things that are right on top of each other so that you couldn't draw, let's say, a second line out from this one, okay, where it was just going to be too close, what you do instead is draw down and then over and then make your label. Everybody follow me there? But at the end, it needs to be attached to a horizontal line. It's just a thing that makes it look neat, okay, and organized. Big thing, if you're taking art, they always tell you about shading, because that's how you give texture and dimension and stuff like that, as I understand it, to drawings. You don't shade on a lab drawing. All right? The reason you don't shade on a lab drawing is if I decide that, well, this part's darker than the others, I'll just shade it. What have I done? Yeah, I've obliterated something on my lab drawing. All right? So we don't shade, we stipple. How many people know what that is? Okay, it's going to sound like woodpeckers on crack in the lab because this is stippling. Okay, that's really dark. This would be less dark. So the more dots you put in, the darker it is. The less dots you put in, the lighter it is. That's stippling. All right, but while you're drawing your lab drawings, everyone's going to be going in there. Okay, yeah, and it's crazy. All right. Everybody with me so far? So no shading, okay? Make sure you don't shade on there. And now in terms of where everything goes. Name and date go in the top right-hand corner. All right, so John Smith, November 12th, 2011, although obviously it isn't that date today. Okay. And then below is where the title goes. That's the thing that's counterintuitive. That's why I got a 0 on it the first time. I was always taught titles go at the top. They don't on a lab drawing. They go under the drawing. All right? So this is a drawing of something from phylum Cestota. Okay, it's a flatworm, and it is at 50 times magnification, and it is approximately 1.5 centimeters in length. All right, so those are the things I read underneath. I give it a title, so Paramecium or Euglena or whatever it is you're looking at. The magnification you viewed it under. So if you looked at it on high power, which is going to be for most things, it'll be 400x. Okay, 400x is the high power magnification, and then you do that size estimation thing that we talked about, where you estimate how many fit across, divide the field of view by that number, and you've got, okay, your size. Everybody follow? That's where everything goes. All right, again, you don't have to be a good artist to do a lab drawing. All right, if I was drawing like a picture of a paramecium, just something like that, and it's got usually oral groove, and it's got cilia, okay, kind of along the outside. Okay, this area is usually very dark. This is lighter. Okay, there's usually like a nucleus in here and a nucleolus, and that's very dark, and this is kind of dark. Okay, but you don't have to be a good artist. I'm not, obviously. Okay, but it just has to generally represent the shape of the organism you're looking at. Right? I am not going to be marking you on your artistic ability. I'm marking you strictly on format, unless, of course, your paramecium looks like a dragon. Okay, then you're just fooling around. All right, so yeah, make sure it generally looks like the thing you looked at. For the amoeba is great because all amoebas look different. They're a blob. And they're very easy to draw as a result. Okay, so that'll be the enjoyable part if you have my artistic ability. All right. So those are the big things, guys, about your lab drawings. The other thing is, don't put in stuff that isn't there. All right, and I know that seems obvious, but when you start learning the parts of the cell, there's a tendency to go, oh well, Mr. Coder said there's supposed to be mitochondria in here. I'd better draw them. If you didn't see them, don't draw them. Okay, because usually some of the structures are not visible through our microscopes. They don't pick up stain or whatever, or they're too small, or you just simply don't recognize them, whatever. If you don't know what it is, okay, draw it, but don't label it. If you can't see it, don't put it on there. Don't just assume it should be there. What if you had like some freak cell and it just didn't have any of those? All right, then you didn't draw an accurate diagram. All right, so even if you know it should have something, if you don't see it or you can't recognize it, you don't put it in. All right. I'll know. You'll actually lose marks if you put stuff in there that I know you couldn't possibly have seen with a microscope. Okay. All right, so that should take care of the lab drawings. So next thing is microscopy lab. All right, so problem we're investigating here, guys, is how does a microscope allow us to see tiny specimens? All right, well, obviously microscopes have what in them that make things bigger? Lenses, right, okay, and what does light do when it goes through lenses? It refracts, which is another word for bends, right? You got to work that somehow, obviously, into your hypothesis, okay? So generate a hypothesis about how a microscope works, okay? That would be like the path of light and what characteristics, size, orientation, color, etc. the images are going to have, all right, as a result of that. So an if and then statement that includes those things, please. And then obviously identify your manipulated, responding, controlled variables. Remember to do that part. Okay, explain why. Now, in this lab, it might be a little bit tricky. You're using the same microscope every time. Hint, hint. You're looking at pretty much everything under the same magnification. Hint, hint. But what's different every time? Nope, because you're going to look at pretty much everything on high power with the exception of one specimen. Okay, so it's not, you're not really manipulating the magnification here, but are we looking at different things? Yeah, okay, you're going to be looking at 4 things. You're going to make what's called a wet mount of the letter E. I know that doesn't sound very exciting, but it's actually really important because it tells you about the orientation of objects under the microscope, right? Because it's the one thing that you know how it's supposed to look, and you'll know whether it's backwards, upside down, right side up, okay, whatever. You'll be looking at the amoeba, the paramecium, and the euglena. So 4 things. Okay. Should they all have, hopefully, similar characteristics when put under the microscope? And not by similar characteristics, I don't mean look the same. Should they all appear bigger? Okay, like should their image all be affected the same way? Yes, that's what we're kind of looking for here. So we're manipulating a bunch of different things to see if the same thing happens to all of them. You guys follow me there? So I just gave away the manipulated variable, right? The things you're looking at. Right? Responding variable I also kind of gave away, but I'm not repeating it. Okay, you can watch this later if you want to. Okay, so and again, your hypothesis needs to talk about how a microscope works. That would be the if part. Okay, the and part would talk about, okay, what you're doing in the lab. The then part would talk about characteristics, size, orientation, color, etc. All right, that would be how your hypothesis should work. All right, procedure is pretty straightforward. You'll acquire a microscope when we go across to the lab. I'll show you where those are. Okay, get the necessary slides. Again, they're all going to be on the front table in labeled containers. Take only one at a time, please. All right, there's no need for you to take 4 slides back to your table. Inevitably, that's how they get knocked off the table and broken. When you're done with the amoeba, come up and get something else. While viewing the slides, make lab drawings of the letter E, the paramecium, the euglena, and the amoeba. You don't necessarily need to make good lab drawings today. In fact, what a lot of people have been doing is something very smart. Take your phone, take the camera, Put it against the microscope once you have it focused and move it. It does take a little bit of fiddling, okay, until you get the image to show up in the camera, then snap a picture of it. Now you can take it home and do a really good copy of the diagram whenever you want. I mean, if you want to do a rough drawing while you're looking at it, that's fine, but then you're always going back, and then you're looking at your paper, and you're looking through the microscope, and you're looking at the paper. All right, it kind of is a pain in the neck. So you might just want to snap a picture of it and then just, okay, well, I can draw. All right, look at it on your phone that way. It does work pretty well once you get the angle on the camera right, okay, on the microscope, but it does work pretty good. Then you're going to make the wet mount of the letter E. So making a wet mount, okay. Hey, I drew that. That wasn't bad. I must have taken my time on that one. All right, sorry to give you that. Okay, so for the letter E, all right, when you're making a wet mount, here's what you do. You're going to take a blank slide, which is just a rectangular piece of glass like this green thing here, and you're going to lay it flat. Okay, lay it flat on the table, and you're going to cut out a lowercase letter E from a regular print size like article from a newspaper. Right? There's a newspaper sitting up on the front table and some scissors. You'll just cut out from an article a small lowercase letter E. You'll place that on the slide. Obviously, it's not that thick. I'm exaggerating. Okay? You'll place it on the slide and make sure when you put it on there that it's right side up as you look at it. Then you'll add a drop of water to that with an eyedropper. As soon as you do that, it's going to flip over, okay? And it's going to turn. It's going to do all kinds of rotten things to you that you don't want it to do. That's fine, right? What you're going to do then is take like a probe from one of the dissecting kits that's up front, and you can just push it down until it soaks up enough water that it sinks, and then you can orient it the way you want, okay? Once it's oriented the way you want, then you'll take a plastic cover slip, which is just a square piece of plastic, and you'll set it on the microscope slide at an angle, and then let it go. And it'll drop down onto the slide and squish the water flat. Okay? And the E will stay right where it is. And the cover slip will stay on. Right? Why will the cover slip stay on? Because water is a polar molecule. Think about that. Okay, might be important later. So yeah, it'll stick on there. Okay, really good. Won't come off, and it keeps you from getting the lenses wet. That's the purpose of a cover slip. Okay, if lots of times the lens will actually touch the slide, and obviously if you didn't have a cover slip on, then it would get goo all over it all the time. We don't want that. All right, so that's the purpose of the cover slip. It's transparent. It won't affect anything. So when you put your slide then in the microscope, make sure that the slide and the letter E are oriented this way. So if you're looking at the microscope, you put it in and slide it in so that it was right side up to you. It's not going to look that way in the microscope. Everybody follow? Draw the lab drawing as you see it through the microscope. All right? If I see a right side up letter E, you're going to lose marks because that's not how it looks under the microscope. Everybody follow? Everyone's good with that? Okay. The lab drawing of the letter E will be done on scanning power. Not scamming, scanning power. It is the only lab drawing you will do on scanning power. The rest will all be done under high power because you won't see enough detail on scanning power to draw anything for the others. All right, everyone follow how to make a letter E wet mount? That's the one you should do first. After that, it doesn't really matter what order you look at them in. Okay, the amoeba and the paramecium are easier to find than the euglena. The euglena is quite small. You may want to save it for last after you've practiced up a little bit. Give you some idea what everything looks like so you know what you're looking for. All right, up here, this is a euglena in the top left. It's obviously much smaller than this when you look at it. In fact, on the scanning power, when you first put that slide in, if you have it properly focused, they will look like small blue slivers. All right, that's how small they are, small blue slivers when you get that under scanning power. All right, you may even have to use the high power or the low power lens, kind of like a scanning lens, and get one on the end of the pointer. They're that small, right? But definitely draw it on high power once you get it focused. This is the paramecium. Paramecium is kind of oblong in shape, similar to the euglena, except the euglena has the one single tail, whereas the paramecium have cilia. And I don't know if you can see it really well here, but you can see kind of a blurred edge. Here you can actually see them quite well on this one on the right-hand side. These are cilia. They look like hair, but they're not. Okay, and that's how the paramecium moves. It waves these hair in kind of unison, and it can propel itself very quickly. And this down here in the bottom left is the amoeba. Your amoeba will look generally like this, but not have this exact shape because amoebas change their shape all the time. They're a blob. Right? They surround their food and then just engulf it. Right? And that's what these are kind of doing. Like this here would close around a piece of food and engulf it, and it's called phagocytosis. Okay? To engulf something and eat it. All right, those are the other three things you're looking at. Everybody with me? If you're having trouble getting something focused, let me know and I'll come and help you. Okay, that's no problem. Big thing is when you get to the high power lens, it's going to appear to get darker, all right, because you have a smaller aperture through which the light comes through. So less light comes in and it won't look as bright. You may just need to adjust the light level, okay, with the little rheostat right here. Okay, that will allow more or less light in. The other way to let more or less light in, because we didn't get to it yesterday, okay, on the bottom, underneath the slide here, you'll see this little tab. Everyone see what I'm pointing at? Okay, opening and closing that opens and closes an iris that is underneath the stage, and that controls how much of the light from the light source gets through. All right, if it's closed completely, obviously, well, it's not closed completely, but it's closed most of the way, not much light gets through. If it's open all the way, obviously a lot of the light gets through. The advantage of this is as you play with this, you can increase the contrast that you can see, and if you have something that's not stained, sometimes stuff just suddenly jumps out and appears three-dimensional, and you see detail very clearly by changing the amount of light that comes through. So you can play around with that a little bit as well if you're having trouble seeing things. All right, everybody follow me on that? Okay. So you don't have to necessarily have anything written down before we go across. Just know that that we'll be doing this kind of stuff over there. The analysis questions we'll do after the fact, but you might want to have them in mind while you're working over there. And then same as always, conclusion and evaluation. All right. Again, size estimation. I'll remember always go in the longest dimension. All right. So if we were looking at whatever these are, we would have this would be the longest dimension, and I would say that 1, 2, 3, 4, 5, 6 fit across the field of view. 400 over 6. Yeah. Any questions before we go over there? So we're going to the bio lab this time, not straight across the hall, but down one. All right, just past the boys' bathroom. Okay, let's go.